going to bind. We saw that placement of that promoter tells RNA polymerase where the gene starts, which gene is, or which strand is which in terms of coding versus template strand. So it plays a lot of important roles. Once RNA polymerase binds, it starts to open up the DNA, right? It pulls it apart, which is why probably those promoter sequences are AT-rich, and then starts the process of transcription, reading the template strand of DNA and making a complementary strand out of RNA. We saw that once that starts, sigma has done its job, so it leaves, basically. And we go through a similar cycle for RNA production as we did for DNA replication before. It reads the template strand, it grabs an RNA, complementary RNA nucleotide, puts it in place, makes the phosphodiester bond, checks it, moves to the next letter. And all of that is being done by that single enzyme. So we don't need primase, we don't need helicase or topoisomerase or any of those extra proteins that we did for DNA replication. And then we looked at two types of terminations. So how does it know where the gene ends? We saw that there is some kind of terminator sequence involved. One of them also involves an extra protein, so this is kind of an exception to the thing I just said. Some genes require an extra protein called Rho in order to stop transcription. Rho is looking for that rut sequence. And we looked at a model to kind of explain it. Basically, it seems like Rho kind of scans along the RNA and looking for that rut sequence. When it finds it, it somehow interacts with the RNA polymerase and basically tells it we're done. And then we looked at Rho independent, so this does not need Rho. And we saw a different terminator sequence that after it's transcribed forms a hairpin. And apparently there are various sizes, but the general idea is it forms a hairpin. And that hairpin, there we go, interacts again with RNA polymerase and basically tells it transcription is over. And then it kind of everything falls off the gene. And then we looked at, and then we kind of moved into eukaryotic cells because a few extra things happen there that we need to talk about, one of which are those introns. So we started talking about non-coding sections of genes, and most eukaryotic genes have them. I think the average is like 8 or so per gene, although we saw some have just one, some have many, many more, but a good ballpark average is around 8 per gene. And these get spliced out, as we saw, and the exons, the parts of the gene that actually code for the protein, get spliced together. So this is the first type of post-transcriptional modification. So these, again, in this picture, these blue introns get removed, the other exons get spliced together, and we saw that that requires special sequences at the sort of junctions or the borders between exons and introns on either side. And those special sequences are found and bound to and worked with by RNA protein complexes called SNRPs that aggregate together into spliceosomes. And they have the job of recognizing the sequence, making sure they cut in exactly the right space on either side, getting rid of the intron and gluing together essentially the exons. And I think that's where we got to last time. Okay, so how are we feeling about the process of transcription, initiation, elongation, termination, and so far these ideas about introns and intron splicing? Feeling super, right? Because we're all high on candy from yesterday. Okay. Okay. A couple more things about these introns and exons. Sometimes not all the exons are kept in the transcript. And this has led to the discovery of something called differential splicing. So if each of these blue little boxes represents various exons, usually this is a tissue-specific kind of phenomenon, so this gene for this protein called tropomyosin is found in a number of different cell types, but it's made in slightly different ways, the final protein that is, in the different cell types, because sometimes, for example, if we look at the first two, in striated muscle versus smooth muscle, the striated muscle removes that second intron and keeps the third, whereas in smooth muscle does the opposite. It keeps the second exon, I should have said exon before, and gets rid of the third. And fourth, fifth, sixth, seventh, eighth, ninth, 10th are the same, but then striated muscle keeps the 11th exon, but the smooth muscle does not. And then the 12th exon is kept in the smooth muscle, but not in striated. So what this creates are slightly different variations of the protein that are usually called isoforms. My analogy for this is usually shirts. Okay? So we're all wearing shirts, but we're not all wearing the exact same shirt. Right? Mine is a button-down and has collars. Yours is more of a sweater than a shirt, but you get the idea. There's no buttons down the front, there's no collar, but we would still recognize that as essentially a shirt. Okay, that's the idea of these isoforms as a result of differential splicing. These are all the same protein, it's all tropomyosin, just slightly different varieties of tropomyosin, okay, that we can get from one gene. That's the other critical idea here. So we can get these different isoforms from the same gene. And if you follow the film project, most scientists were pretty surprised when it seems that we can make a human out of only maybe 25 or 30,000 genes, which was much, much lower than anybody had initially predicted. But once we throw in this sort of story, this might begin to explain how we can get something as complicated as humans from maybe 30,000 genes. So in this example, this one gene gives 5 slightly different products, or 5 different varieties of this one product off of one gene. And it's becoming pretty clear that most genes work this way. That most genes have the capability of being differentially spliced in different tissues. So each gene really gives us more than one product in that sense. So this might be the explanation, or at least part of it, for why the total number of genes seems so low. But if we're getting 3 or 4 products off of each gene, then that might sort of compensate. So it's an interesting phenomenon. People are still working out exactly how this happens. So how come the spliceosomes in striated muscle, cut the messenger RNA differently than the spliceosomes in the smooth muscle. So there still are certainly questions about how exactly this works, but the overall phenomenon is pretty well established now, and like I said, is shown to be true for a large percentage of genes. So it's an interesting kind of phenomenon. Does that idea kind of make sense? Right, so like I said here, so this version keeps the second exon, but this version doesn't. This version keeps the third one, whereas that one doesn't. So again, both of these are tropomyosin, just slightly different versions of that particular protein. And before we leave introns altogether, I just wanted to mention some recent work that people have been doing to explain why we have these in the first place. Because they seem, at first glance, I think, to be sort of useless. Right? Why would we have parts of a gene that don't actually code for pieces of the protein? Okay? Because think about what we have to do in order to make this happen. Well, first off, they get transcribed, right? Here we go. Right? So that takes time and energy. Then we have to create this elaborate process to splice them out. Right? So we have these sequences had to evolve. These SNRPs and spliceosomes had to evolve. And as we've seen, they have to do their job absolutely perfectly. Right? Because if it doesn't cut there where it's supposed to, if it cuts there and it leaves that extra G in, the rest of the process is screwed up. right? That's that frame shift mutation that we talked about last time. Right? If you leave an extra letter in and we read the code by threes, leaving an extra letter in screws everything else up. That's called a frame shift mutation. So not only do these things have to exist, they have to do their job absolutely perfectly, where you screw everything up. That's a lot of pressure, in a sense. So since most eukaryotic genes have have introns, and the cells have gone to all this elaborate trouble to splice them out, they must be doing something than just sort of sitting there doing nothing. Right? This is a lot of time, a lot of effort, a lot of energy for things that truly do nothing. So people have kind of looked at various aspects of this. I don't want to spend too much time on this. I just want to point out a few of the more important ones. But they've sort of divided the process into some phases and looked at each phase in terms of what it might be doing for the cell. And it turns out that introns actually do have sort of functions to them, even though one of those functions is not to code for information in a protein. And so they certainly don't have that function. But they do seem to have some other functions at different sort of times along their sort of lifespan. And one of them, as we just talked about, might be that they have some role to play in alternative splicing. Okay, so this differential splicing that we just talked about might be in part controlled by the actual placement of these introns. They might have a role to play in the actual process of differential splicing. They might have a role to play in regulating transcription itself. There's some evidence that the part of the sequence of introns has the ability to interact with RNA polymerase to bring it to the gene to allow it to start transcription, for example. And that's up here too, transcription initiation. There's other evidence that the placement of specific introns has a role to play at the other end of it to make sure that that termination of transcription happens accurately. There's other evidence here, genome organization, that the nucleosomes that we talked about, those proteins, the histone proteins, seem to interact. Let me make sure I get this right. Say it. Right. That the nucleosomes, the histone proteins, interact more with exons than with introns, and that might have a role to play in how those nucleosomes are put together. And we already talked about that might play a role in controlling gene expression as well. And then another picture here. Some people have suggested that proteins that interact with some of the introns have a role to play in export of messenger RNAs from the cytoplasm. We kind of talked about that a little bit in terms of proteins earlier, but for eukaryotic cells, the messenger RNA is going to have to get out of the nucleus into the cytoplasm. So there are specific proteins that seem to interact with some of the introns that have a role to play in getting those messages out of the nucleus, okay, where they're going to be translated. We'll get there soon. Others have suggested a role for the introns and these proteins that interact with them that get the message to where it's supposed to be in the cell. Other people have suggested they might have a role to play in how much protein you get off of a specific message. So, without going into too much more detail, they certainly do seem now to have specific and important functions attributed to them. So they really don't seem to be just these sections of DNA that don't seem to code for anything and just sort of sit around. They seem to have other functions that are important to the whole idea of gene expression. So no longer can we think about them as just this sort of useless stretches of nucleotides that we simply cut out, and they never have any other role to play. People have been now finding specific functions for them. So it was inevitable because, again, it doesn't make evolutionary sense just to carry around this extra material that doesn't really do anything. Evolution wouldn't be in any way an advantage. In fact, it would be a disadvantage. So we kind of always sat back and said they must be doing something, but until recently we really didn't know what that was. So these are all relatively recent discoveries. So again, that hopefully again is another good example of where, again, as thick as our textbook is, there's still stuff we're just beginning to figure out. So there will be plenty for all of you if you decide to go to graduate school and get your master's and PhD. There's plenty that we don't know and don't understand. So even within this list, we're only at the beginnings of figuring out exactly how these things work. Okay, so in short, it does seem that introns do have now functions associated with them, just not coding functions. And another piece of evidence that kind of suggested that they would have to have some kind of function is that they're conserved across species. So if you compare the same gene from, say, humans and, say, mice or something like that, you're going to find introns basically in very, very similar positions. So again, if they're conserved like that evolutionarily, that really usually suggests that there's something to it. There's some kind of function to that intron, because if there weren't, there would be no pressure to keep them sort of in the same places across the phylogenetic spectrum. Okay, so at least a little bit on what these introns might be doing. So that's, overall, intron splicing was our first post-transcriptional modification in eukaryotic cells. There are two others. So we've gone from that primary transcript that had both exons and introns in it. Okay, so we spliced them out. So now we typically have a shorter messenger RNA with just exons. The other two modifications happen to the two ends of the transcript. So at the 5' end, as you see here on the screen, 5' end gets an extra G added to it that is usually referred to as a G cap. So that G, that extra G that you see here, is not encoded in the gene. It's an extra one that's added after transcription. So there's a specific there's a separate enzyme that does that. And it's not put onto the transcript in the usual way. So it's not a traditional phosphodiester bond, as you can kind of see here. Instead of passing this nucleotide through its 3' hydroxyl to the 5' phosphate, right? That's a regular phosphodiester bond. They connect the phosphate, so it's a phosphate-phosphate bond. It's a 5' to 5' bond. Okay, so it's very, very different than the other phosphodiester bonds that we have here. So it's a 5' to 5' bond. Two phosphates are attached together. And we think that this sort of protects the 5' end of the messenger RNA from being degraded once it goes out into the cytoplasm. So my analogy is always, it's getting colder out. We're starting to wear hats or caps soon, right? Kind of protects your head from the cold. Same kind of idea here. It protects the front end, the 5' end of the message from being degraded very quickly in the cytoplasm. Okay, so it gets a modification at the 5' end. It also gets a modification at the 3' end. And at the 3' end, most messages get a long series of A's added on a poly-A tail. Again, those A's are not encoded by the gene. There's no stretch of T's in the gene for this. These are added post-transcriptionally by the enzyme poly-A polymerase. And we think this kind of does the same thing at the 3' end. Now these are put on in the regular way. These are normal phosphodiester bonds. And what we think is that it basically protects this end from being destroyed by nucleases. So almost as soon as the messenger RNA leaves the nucleus, goes out into the cytoplasm, it starts getting destroyed, okay, by nucleases. And that is a normal part of cell physiology. You don't want your messenger RNAs to exist forever. Right? You don't want to still be making a protein that you made the messenger RNA for 10 years ago. Right? You want to turn that over. So you turn it over by having this nuclease, the eraser, and start chopping up the message from the 3' end. But these A's don't actually code for anything. So if I start erasing those, does that affect the protein that this will make? No. So that's what we think it does. It sort of extends the life of the message in the cell for a little while. So some of them are very long, so it's not a consistent number. So that's definitely part of what we don't understand yet. So some messenger RNAs get shorter ones, like say 50 A's, others get 150 A's. We don't really understand that yet. Okay, why some get longer tails and some get shorter, and how the cell knows which ones should get longer and which ones should get shorter. So there's definitely a piece to this we still don't really understand. But overall, this seems to be the idea. So if it has to chew through 150 A's first, then that's going to allow this to exist longer, and you might get more copies of the protein made while the poly A tail still exists. So it's not until it starts actually chewing up the message now you can't make protein off of this message anymore. So that's kind of the idea here. So we have 3 major post-transcriptional modifications in eukaryotic cells. Remember, none of this is happening in bacteria. This is only happening in eukaryotic cells. With intron splicing, we have GCAP at the 5' end, we have poly A tail at the 3' end of the message.
A cell can rapidly adjust to environmental changes by regulating several processes that happen after transcription. In RNA processing, a cap and tail are added, introns removed, and the remaining exons spliced together. Alternative splicing of RNA may create different mRNA molecules from the same primary RNA transcript. Passage of mRNA through the nuclear envelope provides additional opportunities for control.
In other words, these 3 things have to happen to that primary transcript before it's allowed out of the nucleus, out into the cytoplasm where translation is going to happen. Okay, so if it doesn't get a cap, or it doesn't get a tail, or the introns aren't spliced out properly, it doesn't get to leave the nucleus. So that's another control level there. And the other I have to show you is a little bit more detailed one about intron splicing. This has narration to it. Okay, so here's the intron that we want to splice out. Here are the 2 exons that will get spliced together. So a whole host of SNRPs get together and they bind to various parts to this. So some of them bind to the junction between the exon and the intron. That other one bound to that A, where that's going to get connected to first to make that lariat, if you remember from Wednesday. So there are actually hundreds of these different SNRPs around. So when a bunch of them get together, we would call this a spliceosome at this point. This is showing it to us in much more detail than we have to worry about, but it's not a bad animation, just to give us an idea. And so it breaks the bond, breaks the phosphodiester bond between the exon and the intron, hooks the intron to that internal A, and then puts the 2 exons together, and that releases the intron as that lariat. And as we've seen, this interaction here might, again, between the SNRPs and the intron, might have roles to play, as we just saw a few slides ago, in terms of controlling gene expression and other kinds of things. So this is not any longer, can we think about it again as just this meaningless piece of DNA. So again, these post-transcriptional modifications have to happen in order for that messenger RNA to leave the nucleus, go out into the cytoplasm to get translated eventually into a protein, and we'll start that shortly. So again, realize we started out in bacteria, right? We looked at the general process of transcription in bacteria, but then for these post-transcriptional modifications, we moved into the eukaryotic cell because that's the only place they happen. Most bacterial cells don't have introns, they don't get G-caps, they don't get poly A tails. Those three things are exclusively for eukaryotic cells. Okay, so that's a little bit about transcription. If you take Dr. Ingersoll's molecular biology of the gene course, you'll get way more detail about this process. There are many things we sort of skipped over or went over very quickly. So there is a lot more to the process, but for this sort of level, I think if you have a good feel for how this is working, I'll be very happy. Okay, feeling okay about the transcriptional process and these post-transcriptional modifications? Okay, so that's our first part, right? We went from DNA to RNA. Okay, so we copied the information from a single gene into a single messenger RNA that now will leave the nucleus, go out into the cytoplasm and be translated, which is the same part of gene expression, right? RNA to protein is called translation. So let's get started on translation.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
Thanks, this is what we covered already.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so we're going to focus here on the translational process. How does the information from that messenger RNA end up making us a protein? And then we'll follow that protein through the ER and Golgi and secretion next week. Okay, so we have the first player in making a protein is that messenger RNA, right? That has the information to make the protein. But we need two other players to do this process. The second player, as we saw in that animation, are a series of small RNAs called transfer RNAs. So these are first transcribed, just like we just learned, right? So there are transfer RNA genes. They are transcribed in exactly the same way we just learned, right? RNA polymerase hops on the promoter, transcribes the transfer RNA gene. They get introns removed. This is a eukaryotic cell. Introns removed. They don't get 5' caps or poly A tails, but other than that, it's a piece of RNA just like any other. But unlike messenger RNAs that encode information for proteins, this is the final product of those genes. Right? This doesn't get translated into a protein. It stays as what we call a transfer RNA. And all transfer RNAs basically have this same basic structure. That's sometimes referred to as sort of a cloverleaf structure, in a sense, which is a good way to kind of look at it, and it's a relatively easy way to draw it, but in reality, but not very accurate in terms of what it really looks like. So it actually doesn't. The two arms aren't usually out like this. The two arms actually kind of fold over each other like this. But I can't draw this very well, so I always draw them like that. So these two arms actually kind of fold in over each other. So this is kind of what it really looks like, but I can't ever draw that. So I draw it like that. But that more extended structure lets us kind of see some of the important parts of the molecule. And there are sort of two really, really big important parts to this molecule. The first is, let's start up here. The first is at the 3' end. That's where the amino acid is going to be attached. So I guess I jumped ahead a little bit. So what do transfer RNAs do, as we saw in that animation, they are going to bring the correct amino acid to the correct location in the growing protein. Where is that amino acid attached? It's attached to that 3' nucleotide of the transfer RNA. We'll see how that happens in just a second. So that's an important part, the 3' end. The other really important part down here, called the anticodon, is really what decodes the information in the message. So I'll just kind of sketch this transfer RNA up here. Again, the 3' end is where the amino acid is going to get attached. Again, we'll see how that happens in a second. And then the 3 nucleotides down here at that bottom loop are called the anticodon. Now, we already studied the genetic code, right? We know that it's a triplet code that are called codons, right? Each triplet is called a codon. This is the anticodon. So, for example, we saw in the genetic code table that GGG codes for glycine. How does it do that? Well, as we'll see, when we kind of put this all together, the transfer RNA that would attach to the GGG codon what would it need to have if it wants to attach to GGG? What would the anti-codon need to be?
CC.
Right, CCC, right? Complementary base pairing. So CCC can attach to GGG, and as it turns out, every transfer RNA that has CCC in its anticodon has glycine attached to it. So every time there is a GGG in the message, a transfer RNA with CCC on the anticodon binds there and brings glycine to that spot in the protein. That's how all this is kind of going to build. So transfer RNA really decodes the genetic code. That's how GGG goes for glycine. It's through a transfer RNA with CCC as the anticodon, and all of those transfer RNAs have glycine attached to them. That's a really clever kind of system. Everybody kind of get that? And we'll put this all together eventually. Okay, so the next question is, how do we get the right amino acid attached to the right transfer RNA? Because theoretically, how many transfer RNAs do you think there are? Well, how many codons are there? 64. Do all of them code for an amino acid, though? No. How many do not? Remember? It's bigger than 0, but smaller than 64. How many stop codons are there? There are 3. Okay, so as we'll see, those stop codons don't have a transfer RNA with them. So there are theoretically 61 different transfer RNAs out there, each with an anticodon. So how do we get the right amino acid with the right transfer RNA? Well, the answer to that is a series of enzymes that are called aminoacyl tRNA synthetases. And there seems to be one of these enzymes for each amino acid. So there are 20 of these enzymes. And as you can see here, they have kind of a complicated active site. So they have, part of the active site is for a specific amino acid. So this, we want to keep with the glycine example. Let's pretend this one is the one for glycine. Okay, so that's a binding site for glycine, and that's the only amino acid that can bind. Okay, so they're specific for an amino acid. Then there's a site for energy. So to hook these two things up is going to cost you an ATP. And then the third site, the third part of the active site, is for transfer RNAs. And these sites, or that part of the active site, is sort of shaped in such a way that only the correct transfer RNA with the correct anticodon can bind in that site. So again, if this is glycine, then the only ones that should bind here are CCC, CCG, CCA, and CCU. That's why I use this example because I remember that. Those are the 4 that code for glycine. All of the other transfer RNAs can't fit in the aminoacyl tRNA synthetase for glycine. So that's how it hooks up the right amino acid with the right transfer RNA. This process is called charging the transfer RNAs. So transfer RNA with an amino acid attached is called a charged transfer RNA. And again, every time you do this, it costs you an ATP. So hooking those two molecules together costs you an ATP. So keep that in mind because we'll want to kind of keep track of how expensive it is to make a protein. Okay, so every amino acid that gets placed in so far costs us an ATP to attach it to its transfer RNA. So all of these are just floating around your cytoplasm. So these enzymes are just floating around. These transfer RNAs are floating around. They get together, and the transfer RNA gets charged with its correct amino acid. Okay, so you have this pool then of charged transfer RNAs. They're floating around your cytoplasm waiting to be used protein, basically. Does that idea make sense of charging transfer RNAs? Okay. If you're really interested in the actual mechanism, what happens first is that the ATP and the amino acid get together first. Okay, so that adds energy to the amino acid, and then the transfer RNA comes in, and the amino acid gets attached to the 3' hydroxyl of that 3' most adenine. It's all. I probably should have mentioned that. Here, the 3' amino, 3' nucleotide is always an A on the transfer RNA. And now we have a charged transfer RNA with its correct amino acid attached. Okay, but again, that has cost you an ATP. Okay, so